This presentation is an overview of the enzyme pyruvate kinase. There have been multiple isozymes of pyruvate kinase discovered, and to refresh our memory, isozymes are enzymes that carry out the same reactions but differ in their amino acid sequences. Pyruvate kinase has been found in all living organisms. In vertebrates, there are 4 tissue-specific isozymes. The L type is found in the liver, R is in the red blood cells, M1 is found in the muscles, heart, and brain, and M2 is seen in fetal tissue during the early weeks of development. Plants contain 2 isozymes, a cytoplasmic and a plastid form of the enzyme. Most bacteria only have 1 form, with the exception of E. coli, which does contain 2. The structure of pyruvate kinase will differ slightly with each isozyme, but it has been found that they all contain approximately 500 amino acid residues and normally have 3 or 4 domains. Most commonly, you'll see the enzyme with 4 domains, where it can be called a tetramer. There is a helical domain located at the N-terminal, as well as a domain containing a beta-alpha baro motif, one containing a beta-baro motif, and a 4th containing an alpha-beta-alpha sandwich. The structure is a structure of the pyruvate kinase enzyme, specifically for an organism that has a very long scientific name, but we can also call it a European rabbit. So this is the structure of a pyruvate kinase enzyme from a European rabbit, and it just provides a general structure of the enzyme. Now, I'm sure we are most familiar with pyruvate kinase due to its role in glycolysis. It catalyzes the final step of glycolysis, where it converts phosphoenolpyruvate into pyruvate. This is an irreversible reaction because the reaction has a negative delta G value, specifically being negative 7.5. This is very important because the fact that it's an irreversible reaction allows this enzyme to serve as a control point for regulating the rate of glycolysis. And we'll talk about the regulation of this enzyme very shortly. It's also important to note that the reaction converting phosphoenolpyruvate into pyruvate will release ATP. The pyruvate kinase enzyme is specifically a transferase, which will transfer a functional group from one molecule to another. Specifically, it's going to be transferring a phosphate group from phosphoenolpyruvate onto ADP to produce ATP, and this is going to require the help of a magnesium cofactor. So as I had mentioned, the reaction using pyruvate kinase enzyme is irreversible and thus serves as a control point for the glycolytic cycle. Because of this, it's capable of experiencing both covalent modifications and allosteric effectors. Although there are different forms of regulation for the different types of isozymes, we are specifically going to look at the L and the M1 type because they are most commonly known. The L type, if you'll remember, comes from the liver, or is found in the liver, and it is going to be inhibited by ATP and alanine. This means that there is a high energy level and that glycolysis is not needed to produce more ATP. The L type is also inhibited by glucagon, which is released with low blood glucose levels and will lead to the phosphorylation of the pyruvate kinase enzyme, inhibiting its activity. This ensures that the liver is not metabolizing the little amount of glucose that is present because it will be needed elsewhere, such as in the brain. This is counteracted by the release of insulin, which will lead to the loss of the phosphate from the pyruvate kinase enzyme and will activate it. Insulin is released when blood glucose levels are high. The L type is also activated in the presence of fructose 1,6-bisphosphate, which is an intermediate seen in earlier steps of glycolysis, and the presence of this substrate will drive the reaction forward. The M1 type shares many commonalities in its regulation with the L type, and if you remember, the M1 type is seen in the muscles, the heart, and the brain. Specifically, it will also be inhibited by ATP, but the most important point is that it will not be affected by phosphorylation. So glucagon and insulin are not going to be able to regulate the M1 type, and this is most likely due to the fact that the M1 type is seen in the brain, and the brain needs high glucose, high energy levels to function. So it makes sense that it's not going to be inhibited by glucagon. The final point of this presentation that we're going to look at is what occurs when there are not satisfactory levels of pyruvate kinase. So a pyruvate kinase deficiency is a genetic metabolic disorder caused by a mutation in the PKLR gene. This gene is found to be active in liver and red blood cells and is responsible for forming pyruvate kinase. So a mutation in the PKLR gene may result in the improper formation of pyruvate kinase that may result in improper functions, or it may not be able to form pyruvate kinase at all. Either way, it will decrease the speed of glycolysis when there is a deficiency in the pyruvate kinase, and this is particularly damaging in cells that lack mitochondria. So this would be the erythrocytes or the red blood cells. Because it's so damaging in this type of cells, pyruvate kinase deficiency is also known as erythrocyte pyruvate kinase deficiency. When red blood cells experience a decrease in the rate of glycolysis, there's going to be a deficient amount of ATP produced. This will lead to cell death, and the red blood cells will actually rupture or go through hemolysis. When they rupture, they're going to be removed from the bloodstream, and this will decrease the amount of red blood cells that are circulating through the bloodstream, leading to hemolytic anemia. And pyruvate kinase deficiency is actually the second leading cause of hemolytic anemia, and it has been identified to be running in 500 families because it is an inherited condition. So the image that you see here is an example of the hemolysis of the red blood cells. So these red blood cells are healthy, they contain their spherical function or their spherical shape, and the ones with the arrows pointing to them are currently going through the process of rupturing, and you can see parts of them are jutting out from that spherical shape. So this concludes the presentation on pyruvate kinase. I hope that you have found it informative and that you've learned something new about this enzyme.